So when we talk about hyperthermia, most of you might know it, we have a lot of modes of action. So during the last decade, it became clear, besides increasing the blood flow, impacting the vessel permeability, impacting the heat shock response, the membrane, the DNA repair mechanism. Another mode of action is that we have an immune modulation. This is more or less obvious because you know this from higher temperatures like fever, the immune system gets activated, but here from local hyperthermia as well, because you might get release of danger signal and thereby an immune activation. But when we now go for combined therapies, you see, for example, for radiotherapy, we also learned that it's not only a local treatment, that it's also an immune modulator. But on the one side, you know from the radiotherapy, you can increase the immunogenicity of the tumor. On the other side, you have immune suppressive effects, like the release of TGF-beta or the expression of immune suppressive checkpoint molecules on the tumor cell surface. And as often in research with radiotherapy and hyperthermia, you can compare it now with hyperthermia. What does hyperthermia? And I just brought you here two examples. So one example you see here down, it's the heat shock protein 70. And you see that it's increased after radiotherapy in hyperthermia. But when you look in experimental settings, it's only released far out of the cell in the microenvironment when you add hyperthermia to radiotherapy. So this is one example how you increase the immunogenicity by hyperthermia, because you get a release of a danger signal which is normally located as chaperone inside the cell, but outside it activates the immune system. But on the other side, you know this immunological synapse, we have a lot of immune suppressive checkpoint molecules. And I apologize, this is very small, but this heat map, let's say, should only show you that when you combine different temperatures, 39, 41, 44, with radiotherapy, you have a distinct pattern of modulation of the suppressive immune checkpoint molecules. So meaning when we compare it now to radiotherapy, we have with hyperthermia also both sides. On the one side, combined with radiotherapy and immune activation, but on the other side also an immune suppression. And this is now the rationale why we set the theme of our symposium. Could it be another pillar in radioimmunotherapies? Because obviously it would be very clever to combine it with an additional immune therapy to take away the suppressive events. And this was already done preclinically. This is a nice work from Oy et al. She showed very nicely when you combine hyperthermia with radiotherapy and immune checkpoint inhibition. So in this case, anti-PD-L1 and anti-CTLA4, it's the downer line there. You got a tumor growth retardation, so you really got a good anti-tumor tumor immune response by this triple treatment, and this was dependent on T cells. So this is now some preclinical hint that if you combine radiotherapy with hyperthermia and immune checkpoint modulation, let's say, that you might get beneficial responses.